this one for science communication. Now, just a few housekeeping rules before we move on. Please be professional and avoid disruptive or offensive behavior. Mute your microphone unless the Q&A session is on. Keep your video off for the duration of the talks. And if you feel comfortable turning it on for the Q&A session, please do so. Actively and positively engage during the Q&A session, as well as throughout some polls that we might ask you to participate in. And also comply with the copyright and privacy of our speakers. This is an effort that could not be done without a team. So I please ask you to send a virtual applause for the organizers of this webinar series. It's a combination of environmental, analytical, and inorganic chemists that I'm very fortunate to work with. So the idea of having or hosting this type of events is to gather global perspectives from diverse speakers on the science behind PFAS detection, monitoring, and analysis. Just to have also an idea of how we evaluate detection and removal methods, raise awareness among the general public as well, and forecast the future based on what regulations we have currently and what can be done to foresee a better future. So for the agenda of today, we have first Dr. Diana Aga's talk, then that will be followed by a Q&A session. Then we'll have Dr. Christopher Higgins' talk, followed by a Q&A session and some final remarks. But before we get to that, we would like to interact with this poll. So it gives us a sense of how much you are familiar with PFAS and some of the concepts that will be touched during this talk series. So please take the next 30 seconds to do so. And at the end of the session, we will discuss the right answers. Thank you. Okay, the poll is going to be closed in 10 seconds. Don't miss your chance. All right, thank you for those who participated. Now I'm going to pass the baton to Mindy.
Yes, thank you, Carla. So now let's see who are the two speakers for today's session. As you can see here, they're going to be Dr. Diana Aga and Dr. Christopher Higgins. So the first speaker going to be Dr. Aga, who is a Henry Woodburn Chair and SUNY Distinguished Professor in the Chemistry Department at University at Buffalo. And also she is the Director of Reneve Institute at UB. When talking about her research, she is an expert in developing trace analytical methods for organic contaminants in complex environmental matrices based on chromatography and mass spectrometry. And she is also interested in advancing non-target analysis of unknown contaminants in the environment, especially in identifying degradation products and novel forms of PFAS. When talking about awards and honors, she has received an NSF Career Award, Humboldt Research Fellowship, Fulbright's Fellowship, and also she is an ACS Fellow, ACS OGO Agro, and has received Jacobs Kolkoff Medal in 2017. And also she has received SUNY Chancellor's Award for Excellence in Scholarship and Creative Activities in 2019. And these are few from her long list. And when it comes to our second speaker, which is Dr. Higgins, who is a University Distinguished Professor in Civil and Environmental Engineering at Colorado School of Mines. when talking about his research, so he's dedicated to unraveling the intricate dynamics of contaminant movements within the environment, and also his focuses on comprehending the fate and transport of chemicals in both natural and engineered systems. And also his passion lies in investigating PFAS, which illuminates their behavior and impact on our ecosystems. And further, Dr. Higgins is prolific author, boasting more than 130 peer-reviewed publications that underscore his unwavering commitment to this field. And talking about awards and honors, he's honorary professor at the University of Queensland, Australia, and also has received the awards Strategic Environmental Research and Development Program Project of the Year. And also he has been awarded Walter Huber Civil Engineering Research Prize from American Society of Civil Engineers in 2019. So with that introduction, let's invite our first speaker, Dr. Diana Aga, for her precious talk.
Well, thank you, Mindula, and thank you, Carla, for organizing this webinar. I'm going to share my slides. Okay. I know how to do this. There you go. Can you see my slides? Yeah, okay, good.
Yes.
All right, so I have this title that everyone is interested in knowing, whether we can remove forever in the forever chemicals. So it's a pleasure for me to be here and share with you some of the different things that we are doing in the AGA lab in the at the University of Buffalo. So for those of you, I think most of you are familiar with forever chemicals, which are basically perfluoroalkyl substances and are called forever chemicals because of the very strong carbon and fluorine bond. And there are many ways you can classify them. You can classify PFAS as legacy PFAS or emerging PFAS, depending on whether they are the ones that have been already banned, like the production and use, like the perfluorooctane sulfonic acid, in the PFOA, which are the most studied of all the PFAS, perfluorooctane octanoic acid. And so they vary in the head groups with sulfonic acid or carboxylic acid, but they also vary in length. So these emerging PFAS are typically shorter and also more degradable because they have introduced an ether bond to make them more degradable. You can also classify them based on their head groups. So here they are shown as carboxylic acids, sulfonic acids as ethers, sulfonamides, or fluorotelomers. There are more than 12,000 PFAS species in the EPA master list, and it's increasing as every time you check. There are many uses of PFAS. These are synthetic chemicals that have a wide range of applications, from automobiles to electronics, firefighting agents, food processing, packaging, and so on. And because of all those, so you've seen them, I think you've heard them being used in packaging materials for microwavable popcorn and burgers, and most importantly, you've heard a lot of them being used in nonstick pans. And because of this wide use, they're also widely distributed in the environment, and there's been reported by the Center for Disease Control that more than 98% of Americans have some amount of PFAS in their blood. And this is concerning because of the effects that had been associated with PFAS in humans. For example, PFAS exposure have led to elevated cholesterol levels, decrease in vaccine response, decrease in infant weights, and of course, cancers. Because of the wide applications and wide use of PFAS, it has been in the environment, and it has in various sources, right? It's found in industrial effluent from the ones that are manufacturing these chemicals. It's found in areas where firefighting activities had been done. And of course, wastewater and biosolids are a main source. And this is actually one of the things that my group has been involved, is analyzing and looking at wastewater and biosolids. More recently, this year, there was a U.S. Geological Survey that reported that PFAS is found in more than 50%, 45% in tap water in the U.S. So that's quite concerning. In addition, because in the wastewater treatment, the PFAS partitions between the water and the biosolids. So you can find them not only in the wastewater effluent, but also in the biosolids that are applied in agricultural areas. So there are about, at least in 2021, 4.5 million dry metric tons of biosolids were generated in the US. And 43% of these are land applied. And 25% of this land applied, which is about 1.15 million dry metric tons, are applied to agricultural lands, which are concerning because these are the same land where we plant our crops that are used for feeding animals or feeding people. So there has been some report that there's a significant plant uptake of PFAS, and that's something that people are concerned about. So the title of my talk is whether we can remove forever chemicals and making them no longer forever. So there are many people looking at different ways of removing them. And I'm only going to focus on the things that our group has been doing. And what the first group, or the first way, is to do nanohybrid materials, basically a combination of nanotechnology and also biodegradation with mixed bacteria. So we're also analyzing, or we're examining anaerobic and aerobic bacteria in wastewater, and also using activated sludge or membrane bioreactor. I'm going to focus on these two because our work on anaerobic membrane bioreactor is still preliminary, but we are seeing some promising results in the use of nanohybrid nanomaterials and also in aerobic bacterial degradation. So the problem with the PFAS is that biodegradation is not very effective. Sorry. Oops, sorry, I'm going backward. There you go. It has been shown that when there is a carbon that is not fluorinated, that's actually faster when you do biodegradation. So in our preliminary work a few years back, we have found that nanohybrid materials can degrade partially PFAS to short chain. So we had this idea that if we combine nanotechnology and bacteria, we can degrade completely PFAS. So that's been actually some of the bulk of our work, where we're using redox active nanohybrids to catalyze degradation of PFAS, whether partially or completely. But if they are partially degraded, we can combine them with microbial communities for complete biodegradation. This work is funded by the National Institute of Health, and we have some preliminary data that I can share with you. So the graphene oxide nano zero valent iron nanohybrid materials is the one thing that we've been looking at, where we incorporate this zero valent iron to graphene oxide, and we look at least we start with two of the PFAS, PFOA and PFOS, and we found that they're actually degraded into short chain PFAS. And we can do this by several experiments using LCMS for characterizing the byproducts, and also using ion chromatography to determine the generation of fluorides, because of course, degradation of this will mean defluorination or removal of fluorine. Because I only have 20 minutes, I'll just give you the main results from this data so far that we've been working on this project for a couple of years now. We found that these nanohybrids with this graphene oxide can actually remove PFAS and PFOA. Here we look at degradation from 5 minutes to 20 minutes, and we see that in PFAS, we can actually see more than 80% removal for low concentrations of PFOA at all the way to 50 ppm, also from 0.4 to 50 ppm. Sorry, don't know why I did that. Don't know why it's going down, sorry. But then with PFOA, there's a little bit of a different observations where at higher concentration, we see a decrease in removal. So that's at least promising because anyway, in the environment, at least in wastewater, we would see lower concentrations. We haven't really seen up to 50 ppm in the typical wastewater. So at low concentration, we actually can remove them at more than 80%. So that's very promising. To do this, we have to make sure that we not only measure the decrease in the PFOA or the compound that we put into the bioreactor or into the system, but we want to be able to see the generation of fluoride. So here we use ion chromatography to measure defluorination and also the formation of acetate and formate, the defluorinated short chain PFAS. So we're happy to say that we could actually see these chemicals and as an additional proof to show that there is in fact removal and degradation. So here are some of the data that we can show. These are initial concentrations of PFOA. As you increase the PFOA removal, you can also generate increased acetate and also formate. So these are the defluorinated short chain PFAS. So again, we can measure fluoride generation as an additional evidence that we are degrading these chemicals. So this work has just been submitted to ESNT. It should be not in preparation, but submitted, and we're waiting for the review. The next thing I want to talk about is the anaerobic degradation and aerobic degradation of bacteria. So there's been a lot of report that anaerobic conditions using this inoculum of the hollow cocoids KB1 has been shown to degrade PFAS, but also we want to see mixed cultures from actual wastewater treatment plants. In wastewater treatment plants, there are some different wastewater. So you can see a mixed culture or mixed activated sludge. There's also, at least in Buffalo, there is a 2-stage activated sludge where it has a shorter sludge retention time and a longer sludge retention time for nitrification. So we've used some of these actual wastewater to determine whether we can transform PFOA. And of course, we will always use LCMS to determine the degradation products and ion selective electrodes to determine fluoride production. There are precedents of the published papers that show that anaerobic bacteria can actually remove PFAS and PFOA, not completely, but at least these are some of the papers that have shown this microbial species being able to degrade PFAS. In our work, we actually use this KB1 enriched culture in anaerobic systems, but unfortunately, this system did not show removal of PFOA and PFOS. So we went immediately to anaerobic bacteria because we have also shown from literature that there are several bacteria that have been isolated by different literature showing there is removal of FTOH. So it's because of this work, we thought we should make it into aerobic culture. So we use these 3 different systems. We have mixed sludge, we have the short retention time activated sludge, and the long retention time nitrifying activated sludge. We use these 3 test compounds for starting point, and we incubated them from day 0 to 21. And we found degradation of PFAS and PFOA. So that's also very promising in all systems at different rates. Also, we have seen different rates of degradation from the different PFAS that we used. So one interesting thing is that we could see changes in formation of short chain PFAS from all these systems, from this test system. So we can see from PFOS, which is 8 chain, going to 7, 6, 5, and 4 chain PFOA. We've been able to analyze this by LCMS, but you might wonder, does it go to 3 carbon and 2 carbon and so on? Our method at this time is unable to determine the shorter than 4 chain, although we are developing a method now to be able to detect the shorter chain, ultra short chain PFAS that can analyze for C3 and C2. So that should be something that we will look at. The one thing that we have also observed is that PFOA goes all the way to TFA, so this trifluoroacetic acid, which is just a 2 carbon PFAS, which is not very easy to capture in the typical C18 column with LCMS, but we have seen this using nuclear magnetic resonance NMR with fluorine NMR, and I will show you in the next few slides. But in ion chromatography, we can observe formation of acetate and formate. And of course, as I mentioned, the fluoride formation in all 3 systems. So yes, we are able to degrade PFAS in at least in the lab systems that we are observing. I want to switch quickly before I run out of time to the other work that we have been doing. So I discussed removal in liquid, but of course I mentioned there's also a lot of PFAS in biosolids, the solid part in the wastewater. Our group have submitted, have a paper recently published in Journal of Hazardous Materials Letters, where we show that biosolids can be transformed using pyrolysis. So pyrolysis and gasification are one of the major technology that is being used in treatment of biosolids before they are applied in the field. But the question of whether PFAS are removed during this system is still not quite clear. So we have some preliminary data where we actually sampled full-scale pyrolysis treatment plant and took some of the samples from various points. So this is a schematic diagram of the pyrolysis system where we collected samples. We collected dried biosolids, which is the feed material before it is pyrolyzed. And the product of pyrolysis is a biochar, which is eventually applied in the field. So we collected those. And then there are also these condensates, which we call pie oil and pie tar. This picture here is how they looked. They're not very easy to analyze. And so therefore, for LCMS, is going to be very challenging. So we did the total fluorine instead using the combustion ion chromatography, where we could measure the fluoride ion and also the total organofluorine by some extraction to differentiate between the fluoride ion and the organofluorine. But you can see here the different samples that we collected from biosolids, pellets, that is before treatment, and then the biochar after pyrolysis, and the pie-tar and the pie oil. So you can see there's actually a high amount of fluoride ion in the biochar. So you can see it's not completely removed during pyrolysis, and also during the condensate. So what we did is we washed the biosolids to remove the dissolved fluoride ion so that we can determine what is extractable fluoride and what's the non-extractable organofluorine, which is the bottom line here. So you can see that there's a lot of non-extractable organofluorine in the biosolids and also in the biochar, that is the byproduct that's being applied. So it appears that the pyrolysis does not remove completely the organofluorines. But in what form they are, we don't quite know yet. So we have this preliminary data on what we determine in the biosolids, in the biochar, where you can see the biochar actually has a lot of PFBA. This is the 4-chain PFAS, and also PFMPA, which is an ether PFAS, which is lower in the biochar compared to the biosolids. But then there is this lower concentrations of different types of PFAS, which are, when we looked at, so we said there's a lot of the 4-chain PFBS, and the 6,2 FTS is also a lot in the biochar. And this is just one sample. So what we wanted to see is, what about the short chain trifluoroacetic acid, which we cannot see currently in our LCMS method. So fluorine NMR is a good complementary technique, because fluorine NMR, as we've shown in our previous publication, does not actually need to be cleaned up. You can basically extract this biochar and have a very dark sample and use it. You don't need to clean up. There's no ionization suppression. So you can see specific signals from the CF3 terminal group at this about 82 ppm shift. And it's very far away from the aromatic fluorine, which is what we use for our internal standard as a reference standard. And then you can see that the trifluoroacetic acid also is separated from the long chain PFAS. So it is a good way to detect byproducts if it's in high concentration. And what we see from the biochar is that there is about 11.2 nanomoles per gram in the biochar. And in the pie-tar, there is higher amount of total, we say total PFAS, because we with this NMR, we cannot distinguish what type of PFAS it is. We can just say it's the about total PFAS is 1225 millimoles. So to give you some time for questions, can we remove forever in the forever chemicals with all these different things that we're doing? I am very optimistic, and I would like to say, yes, we can, at least in the lab scale. We haven't done it in full scale. So at least in the lab, there's a lot of potential. There's a lot of promising technology that we can explore. It is yet to be seen in the field, and that's something that we have to do with collaborators who are actually doing some of the full-scale remediation. So with that, I'd like to thank our collaborators from the various funding support that we got from NSF, EPA, NIH. And of course, all this work that I presented, you will recognize some of those slides from many of my students. So Josh, and actually I shouldn't name everyone here has contributed really, from the methods development to extraction to analysis and preparing the slides. So thank you, everyone. And I'd like to see if I could answer some questions.
Thank you so much, Dr. Raga, for your very informative and insightful talk. So the room is open to questions. If there are any questions, then you can just unmute yourself or type it in the chat box. Okay, there were also like a few questions which were submitted during the like registration process. So one of them was like, what are the main challenges in removing PFAS from wastewater and biosolids, and how do they differ from other contaminants?
Well, there's a lot of challenges, right? One of the challenges is typically people measure removal by measuring the initial concentration of the known PFAS and looking at whether they are there or not after treatment. But often some of them are just transformed. So as I shown you in my presentation, we can target the transformation products if we can predict what they are. But sometimes we don't know what the transformation products are. And if we don't have the standards, we cannot necessarily measure the byproducts and the toxicity and the importance of the byproducts. And because there are limited number of standards, quantification of them is also a challenge for those that are unknown. Thank you for the question.
So much. I think now we can move forward to Dr. Christopher again. Stop.
So thank you.
Good afternoon, everybody. It is just afternoon here, but I will go ahead and get started. And that was a great talk by Diana, and I will be talking about something different, not any treatment technologies, but more kind of fate behavior. So I really appreciated that perspective. Let me get here. Okay, I think you guys can all see my screen now. Give a thumbs up. Okay. Still have to ask that question after how many years of Zoom, right? So what I'm going to talk about today is some of the challenges of dealing with these compounds, these poly and perfluoroalkyl substances or PFASs as environmental contaminants. I'm going to give you kind of a soil perspective. So we're not really talking about treatment, but really about behavior and what it might mean ultimately for human exposure to these compounds. So I like to start my slide with thanking all the various folks. There's a number of folks whose work I'm going to talk about. I'll try to highlight the individuals when they come up. It's not quite rafting weather here in Colorado, but this is what we do like to do in our neck of the world when the weather's nice. It's actually probably going to be about 70 today, so it'll be a nice day. So jumping right in, we've heard about PFASs already from Diana. I'm going to focus on a really important distinction that I see with these chemicals that I've been working on for a number of years. And that is the difference between perfluorinated compounds and polyfluorinated compounds or polyfluorinated precursors. So, you know, the perfluorinated compounds, I'm speaking to a lot of chemists, I think, all carbon fluorine bonds, no carbon hydrogen bonds. Polyfluorinated compounds, we often refer to them as precursors because they have carbon hydrogen bonds that can be broken over time and under the right conditions to form the perfluoroalkyl acids. And as we've already heard from Diana, once you get to perfluoroalkyl acids, it's very difficult to get them to degrade. It can be done under the right conditions, but often very energy and very specific energy intense and very specific conditions. So there are two classes of two primary classes of polyfluorinated chemicals that can form the perfluoroalkyl acids. It has to do with how they were manufactured or how they've been manufactured. What we have on the left is what was called an electrochemical fluorination type chemical, sulfonamidyl ethanol, versus the right is a fluorotelomere chemical. And these are made via a telomerization process, so adding different building blocks together to get the various chains. But what's important is that both of these are polyfluorinated species or polyfluorinated compounds, and over time they can transform to the perfluoroalkyl acids, whether PFOS on the left or PFOA on the right. And of course, there are longer and shorter chain versions of all of these, so you have to keep that in mind as we're talking about these precursors and the perfluoroalkyl acids that can be formed. So one of the questions I think was great, how many PFASs are there? It's not just about PFOS and PFOA. This was a paper we put out a number of years ago, discovering not just 40 new compounds, but 40 classes or subclasses of PFASs derived from firefighting foam. And any one site that we tend to look at that has used this firefighting foam has typically between 100 and 200 PFASs present. And that's without really looking very hard, to be honest. That's using our standard analyses. So there are sources of PFASs, all sorts of different sources. I'm going to focus a lot of my time on these aqueous film-forming foam, or HBLF releases. And this is material that is used primarily by the military, and it's only 3% of fluorochemical production was for firefighting foam, but because the concentrations are very high, often gram per liter levels, when released to the environment, they can be pretty significant in terms of the levels. So little HBLF goes a long way. 1 5-gallon bucket can contaminate a water supply at the MCL levels of about 100 million people. Or if you have a Olympic-sized swimming pool, that's about 25,000 years worth of drinking water supply for the US population. And the problem with that is that there's a lot of places around the country where this might have been released. So, it doesn't take much to contaminate, and there's a lot of places around the country where it was released. So when I think about PFASs and the challenges that we face related to PFASs in the environment, the first dimension I think about is the chemical composition. So we've talked, there was a question about how many different compounds are out there. Personally, I think PFASs are not the most complex group of compounds. Pharmaceuticals and personal care products, because those are grouped more on their function as opposed to their structure, I think do represent a slightly more complex group. But PFASs are pretty high up there. Certainly more types of structures than we see for BFRs or PCBs, because the tail can be various lengths, and the head group can be various types of compositions. So I think that's one way that they start to be very complex. And so one question I often get is, how many compounds should we worry about? How many compounds should I be analyzing for? A former postdoc of mine, Carrie McDonough, now at Carnegie Mellon, put together a nice paper that summarizes this. And NMR is there, Diana, as a technique to measure. These various ways of measuring these compounds have varying degrees of confidence in identifying them and specific and also quantitative information. So in my particular lab, we tend to focus on things in this section, not just the targeted compounds, but also things that are amenable to oxidation via the top assay, which we won't talk about today. But more importantly, things that are amenable to high resolution mass spectrometry, primarily using LC, which we do a lot of in my lab. And what this means is that when we go out and sample a site that's been impacted by the firefighting foam, this is just an example of the diversity of the structures that we see. A lot of polyfluorinated substances, I'd say that's where the vast majority of the diversity lies, is in these polyfluoroalkyl substances. And again, 100 to 200 at any given site. We are now looking for around 1,400 potential compounds at any specific HBLF impacted site. If you want to do high resolution mass spectrometry work on PFASs, I would point you to the NIST, U.S. National Institutes of Standards and Technology, now maintains a list of accurate masses, and they're working on a library where you can go query your high resolution mass spectrometry data for these types of compounds. And as you see, they've got all these kind of fun structures, which has all sorts of implications with respect to the second dimension of the chemical complexity, and that's the environmental behavior. And primarily because of those head groups, I think that's where, and the fact that they have different chain lengths, that's where I think PFASs really start to distinguish themselves as problematic environmental contaminants. So one of the questions that we often get asked for these sites that are impacted by these firefighting foams is, if these compounds are so mobile, we talk about them being present in groundwater plumes miles away from where they're released. Why are they still present in vadose-sown soils? In other words, why do you still see high concentration of these compounds very close to the point of release? And there's two main components of the kind of the working hypothesis. This is really due to two things. One is the slow release and biotransformation of precursors at these sites. These polyfluorinated compounds are slowly breaking down over time and releasing the perfluoroalkyl acids, which tend to be more mobile. But also the fact that these are air-water interface surfactants. We use them in foam, and so they get stuck at the air-water interfaces in the subsurface, and there's a lot of those in the vadose-sown. I won't have time to talk about that today. There's a lot of really interesting work going on in that regard, but I will talk a little bit about this slow release transformation. So when we go out and look at these HBluff impacted soils, most of the time people are looking for things like PFOS and PFOA because those are the ones that are being regulated primarily, excuse me, in terms of levels in drinking water. But they're not always the highest. And so what's shown here is ranked from left to right, high to low concentrations. I note that some of these are estimated concentrations because we don't have standards for them, but we are trying to get more and more standards of these compounds being made so that we can be truly quantitative in terms of assessing their abundance. And the other thing that's shown on this is the ratio, which is the light gray bar. And that's really just showing that when we're comparing different ways of extracting these compounds, if you don't extract for them, you will miss them. And so, the ratio is basically comparing different methods that Anastasia Nickerson, a former PhD student of mine, employed, basically landing on the need to do both a basic methanolic extraction and an acidic methanolic extraction to really get some of these compounds off the soils. But last but not least, I want to point out that the compounds in red are compounds that you can only see if you look for them in ESI positive ionization mode. So most of the time, people are looking in ESI negative ionization mode, and you'll see a lot of the compounds and some of the precursors. But in these soils, these are compounds that we can only see if we look in ESI positive mode because these are cationic and zwitterionic compounds. And so it just speaks to the fact that there's a lot of PFAS mass sitting in these soils that we need to be potentially paying attention to as these chemicals are potentially transforming over time. So another one of my postdocs, Andrew Meisel, who actually is now at NIST, did a series of studies looking at release of these compounds from the soils. So in other words, we collected soils, brought them back to the lab, the foam had been applied in the field and aged in the field for sometimes 20 or 30 years. How did, if we did a leaching assessment, how do these compounds come off? What compounds actually come off? What is the mass balance of the compounds coming off versus those staying on the soil? So we did all these soil leaching experiments looking at the soil, but also the effluent via high resolution mass spectrometry using LCMS. And these are the sorts of profiles that he observed. So note that it's relative concentration, so C versus C max, but the x-axis here is essentially log pore volume. So the things that are eluting at 1 are taking 1/100th of the amount of time to come off the soil as the things that are eluting at 100 pore volumes. So pore volume and time are essentially linear related. What you see is that the carboxylates come off in a very chain link dependent manner, as do the sulfonates. Sulfonates tend to take a little bit longer to come off the soil. But what's particularly interesting is that when we look at the precursors shown on the bottom, particularly the bottom left, they also come off in a chain link dependent manner. So the C3, C4, C5, C6 versions of that particular class all also elute in a chain link dependent manner, but they tend to elute much later than the perfluoroalkyl acids. Also shown here on the right is essentially holding, so on the left, it's holding the head group constant, modifying the length of the tail. On the right, we hold the tail constant and look at the different head groups. And PFHXS, which is also one that has been proposed for regulation, comes off relatively quickly, but all these other PFHXS precursors sometimes elute much later 100 times later in some cases than the PFH excess does. So we see lots of these different compounds coming off the soils. They're coming off in a chain link dependent manner, but these cationic and zwitterionic compounds are being much more slowly, excuse me, released from the soil. And if we do a mass balance on this, looking at the fraction of mass that is actually eluted, it turns out that a lot of those precursors, these compounds down here on the bottom left, really we don't, even if they're coming off much later, what actually is eluting is a much smaller fraction of the total mass in the system. Of course, we're estimating here again because we don't have standards for all of them. But you know, some of them are slowly coming off, but not much. What's interesting is that the compounds that do come off, they really do swamp the mass of the other compounds there. So again, looking at PFH excess and its precursors coming off the soil, PFH excess is that small sliver down here. It comes off early, but if you look at it relative to all the other compounds in the soil, it is a relatively small fraction of the equivalence of these compounds in the soil. I'll note in particular FHXSA, we have an authentic standard for this. So, you know, we're actually not so much estimating the concentration, we're relatively well quantifying its concentration. And it is a part of a class of compounds, the perfluoroalkylsulfonamides, which are potential precursors to the sulfonates, but also are semi-volatile, which raises all sorts of questions. If these semi-volatile compounds are present in the soils, are present in groundwater, we see them miles down gradient from the source impacted areas. What does that mean in terms of their behavior and eventually human exposure? And that's actually where I'm going to kind of end before I have my kind of final slide, just briefly talking about human exposure. And that's where I think PFASs really do take the cake with respect to environmental issues, is that we are exposed to these compounds in such different manners, and there's not one route of exposure that is always dominant necessarily. Drinking water sometimes can be the most dominant. And so this is a figure that I've adapted slightly from one that Elsie Sunderland from Harvard put out, where it was trying to trace essentially, you know, these are all industrial manufactured chemicals. They get out into the products, they get into consumer products and industrial products, firefighting foam releases into the environment. Eventually, we can have the either direct exposure to the products in our homes, in our carpets, in our clothing, or the indirect exposure through the environment. And you know, it's really easy to measure these in human blood. I think Diana noted the 98%. We just finished a batch of samples where it was 100%. And you know, these are relatively easy to see in human blood. And it's because we're exposed through our drinking water in some cases, mainly food for most people. That's the dominant source of exposure, but you can have consumer product exposure. And what I'm teasing here is this question about these semi-volatile compounds. So if we have these semi-volatile intermediates being formed and present in these soils, you know, does that result in an additional source of exposure that we need to be concerned about if you have a household that has HFLF impacted well water? And there's plenty of households around the country that have that concern. So just to kind of wrap up in terms of talking about challenges for the global community, you know, these compounds are ubiquitous in the global environment. It's really easy to see them pretty much in all sorts of media all over the globe. We're going to have to prioritize what we focus on. And so how do we make decisions about that? I think that's a conversation that probably needs to be had, particularly when it comes to soils and water and things like fish. Compositional chemical complexity is daunting. There's a lot of compounds. You need to pay attention to the chain length and the head group and what can actually convert into what. And so understanding all of that is really important if you're trying to understand exposures, because you could be exposed to a precursor and you don't measure the precursor, you measure the transformation product in someone's blood. And it's something that we have to pay attention to when we're looking at these chemicals. Their fate and transport in soils is complex. You've got transformations happening. You can have the air-water interfacial behavior occurring. You can have, you know, one of the things that has surprised me the most, and I haven't talked about it here, but is there's kinetic limitations, even for some of these compounds, which actually don't absorb that strongly to the soils. You can have kinetic limitations to their behavior, and that's a little bit surprising, but it's something that clearly we're going to have to understand better if we're going to try to make models and ultimately clean up soils. And then this last kind of question, just kind of spurring the idea of, we're trying to understand exposure of these compounds. I think the title of your series is, in the home, how and what does that mean? How do we capture the full exposure of humans to these compounds? I'll add one last thing. An interesting thing on the blood work that we've done is that these sulfonamides that are semi-volatile are also not actually present in serum. And most of the time when we go to measure for human serum, we for PFASs in humans in terms of biomonitoring, we look for them in serum. But we did a study where we compared serum and whole blood using a microsampling device, and we saw for one of the sulfonamides, basically half the people had measurable levels of sulfonamide in the whole blood, and none of them had measurable levels of sulfonamide in the serum. Why that is the case, that's a whole other question. But I think it begs the question of how we're actually characterizing the full extent of exposure to these compounds. With that, I will just point out this is where I live and where I work, and I'm happy to take any questions if you have them.
Thank you so much, Dr. Higgins, for such an interesting talk. So now I'm opening the floor for any questions that you may have. You can type it in the chat box, or feel free to unmute yourself.
Can I ask? Am I allowed to ask questions?
Of course.
Go ahead,
Very
Dan.
interesting, Chris. I have a lot of questions, but I know this is for the students, so I will keep my questions to some limited number. I'll ask two. Well, comment. You're making me jealous of all the pictures you show about Colorado. Very beautiful. The positively charged or the PFAS that you can only determine by positive ESI, are they specific to AAAF, or are they also found in wastewater?
To the best of my knowledge, they are specific to HFF. I have not seen anyone specifically looked in wastewater. I mean, I would look in sludge. You could certainly look in sludge. But I think they were intentionally added as components of the firefighting foams. To the best of my knowledge, none of those chemistries were also used for consumer products, but it is a very complex landscape when it comes to different consumer products. So I wouldn't be totally surprised if a handful of them were actually used in some consumer products.
Okay. And then my last question, unless nobody asks question, I'll keep going. But the NIST database that you mentioned, is that agnostic to the type of data or mass spec you're using? Is it only reporting high resolution mass spec, or do they also put fragmentation when you're using them to identify unknowns?
So the NIST effort, they have two different products that they're working on, and I actually think both are out now. And you can probably Google NIST PFAS GitHub and get access to them. One of them is a mass interest list, which is presumably folks who use high-res mass spec. You collect full spectrum data. You go and look to see, are any of these compounds of interest in my sample. So it's just an XIC list, as we often refer to it as, an interest list, right? The second, and it's agnostic. I think it's basically a CSV file or text file that you can import and use however. The second thing that they've been working on is an MS2 library. And this is also meant to be most used by HRMS systems, but I do think they have some what we call low-res data in there. So if you just had a low-res system, you could also probably use it. It's probably going to be most useful for folks who have high-res fragmentation data and you want to compare spectra. That's honestly I just learned, I think, of it's they've been working on it a while, and I think they finally just went live, so I haven't actually tested it myself, but that is something that I think is now available.
Okay, thank you.
Good. So we have here one question in the chat. Alexander asked, how is Moody fluorine NMR different from traditional fluorine NMR?
Yeah, so we talked a little bit about that in the paper, and if I recall the differentiation, because it's been a while since I've thought of this, I am pretty sure that was a recognition that Cheryl Moody had proposed probably on 20 years ago now, that essentially to do exactly what Diana did and showed works quantitatively, is that if you look at the CF3 group as a specific feature, that you can use that to essentially quantify, because pretty much most of the PFASs that we really care about have that terminal CF3 group, at least one, in their structure. And I think that was what she had proposed, and that's why we differentiated that as compared to integrating the entire fluorine signal to get all of the fluorines. I think that's what Cheryl had proposed.
Interesting. All right. Thank you so much. I think we're done with the Q&A session here. Carla.
Yes, thank you, Jonathan, and thank you so much, Dr. Higgins, for that great talk. So we're going to wrap this up just by showing some of the answers to the poll, and Mindela is going to take it over.
Yeah, so with the talks, I think you have to got the answers for the most of the questions, actually all of the questions. So if you wondered, actually from the EPA reports, they tell there's over 12,000 PFAS species, and here I'm emphasizing the new June, as of June 2022, the EPA health advisories has gone down to PPT levels, as you can see here, for few of the PFASs, the common PFASs actually. So achieving lower LODs as of PPT levels is challenging in today's analytical research field. Although you are going for that low, it's not telling that there's no PFAS at all. So it's just below the LOD. And also those are the kind of answers I think for the poll. If you get wrong or correct, I think with this Zoom, the webinar series, you have a good idea about the PFAS now. So with that, I'm gonna hand over the platform to Carla, saying PFAS are actually everywhere.
Thank you, Mandy. So just to wrap up, and also tell you a little bit about our next webinar. So save the date. We're gonna have Dr. Cannon from the Environmental Health Sciences talk with us next month, November 15, at 11 a.m. Eastern time. So feel free to join us. If you scan that QR code, it will take you to the registration site. But if not, we can follow up with you if you're interested on joining us. You can always get to see what we are up to at our Renew webinar series in PFASfutureLeaders.com. So thank you everyone for joining us, and thank you Dr. Aga and Dr. Higgins for your time and for all your knowledge, and thank you for sharing that. So this is going to go global, hopefully, and everyone's going to be aware of your research. So again,
thank you.
Round of applause. Thank you.
Okay.
Bye everyone. Thank you.
Thank you. Bye.